So now that we have a basic understanding of the cell cycle, we can begin to explore how the cell cycle is controlled. And it, of course, is a molecular control system. It's going to be proteins that are involved with how often a cell divides, right? Now, this is a very complex process, and we're going to look at just one example, the cyclin proteins, to kind of understand how it can be built. But the idea of a cell control system, a cell cycle control system, is that it's going to work like a clock. And that over time, as we move through the different phases, we're going to also have checkpoints that say, are we really ready to go on? Right? Are we done with G1? Are we ready to move into S phase? Are we ready to go into M phase? If we're in M phase, are we really all lined up and ready to split? So these are some kind of critical points that people have derived when they study the cell cycle. So their major checkpoints are going to be, again, the G1 checkpoint. Is the cell big enough? Is the environment favorable? Should we spend the resources to copy our DNA? G2 checkpoint, again, is the cell big enough? Do we have enough resources? Is all the DNA replicated? Is the environment favorable to go into M phase? And then finally, the M phase checkpoint, basically the biggest one there is where are all the chromosomes lined up on the spindle? Because if you don't separate that duplicated chromosome and get each chromatid going in opposite directions, you're going to have daughter cells that are destined to die. You need a complete genome, you need all your DNA for this to work. So this is a really, really important point. So we see these checkpoints at what you would think of as logical ways to check into the system and make sure we're ready to move forward. Now, this introduces a really interesting idea, and an important one, is that all the cells that we've been talking about in the cell cycle are actively dividing. So this is like you cut yourself. Now the cells in that cut area are going to keep dividing, right? Because now they've been kicked on, they need to replace the tissue that was lost, and they're going to keep doing that until they receive a signal not to do that, right? So they're going to exit the cell cycle, and we call that the G0 phase. So that G sub 0 is G0. So a cell is going to be, newly minted from the M phase. This new daughter cell is going to start to kind of gather its resources, and when it reaches the G1 checkpoint, it may get the signal to not go ahead. And so then you just hang out. You do your cell stuff. You're not actively dividing. You're not quote-unquote on the cell cycle anymore. I think that's an important distinction to make. So think about that one. Now, again, what is regulating this clock? What is this clock? It's proteins. And in many cases, it's combinations of protein kinases and phosphatases. So again, reviewing that, remember phosphorylation and dephosphorylation can act as a switch for a protein, turning them on or off, right? Adding that phosphate group or removing it, changing the shape of the protein, allowing it to function or to shut it down. Now, specifically in the cyclin system that we're going to look at here very briefly, we're going to have two proteins that we're going to focus on, the cyclins and the cyclin-dependent kinases, or the Cdks. All right? So the activity of these proteins is going to fluctuate during the cell cycle. We're going to see an increase of the cyclins, and that's going to allow the cyclins and the Cdks to get together to form the MPF. Again, it's a lot of vocabulary. The maturation promoting factor, or the M phase promoting factor, is the MPF. And so I recommend writing all this stuff down and trying to get it in your head, because then you can have a conversation about how it works. So this cyclin CDK complex, the MPF, is what's really going to be our big AOK, the green light to go through the G2 checkpoint into the M phase. So if we graph that, we can look at it like this, and we can see a graph up top of the cell cycle, G1, S, G2, and M, and then underneath the concentration of cyclin over time, or MPF activity. When you kind of map that out in our circle, we can actually see that we've got a CDK population in the cell. And there's going to be some other signal that says when we're synthesizing our DNA towards the end of the S phase that we need to turn on cyclin. So cyclin starts being produced and accumulating, and as it accumulates enough, it can start to interact with CDKs to form enough MPFs that when we reach the G2 checkpoint, that MPF activity says, yes, it's okay to go on. So things that would perturb this would be things that would say, hey, we're not ready. to go into the M phase. So signals from the environment or faulty DNA replication or something that says we're not ready to go on. But if everything is working right and the conditions are right, the MPF should peak as we move into the M phase. And then the cyclin would then be broken down and reset the clock. Again, this is just one example of many different kind of molecular clocks that are going to be involved in regulating the cell cycle. And it can get very complex, but it's fascinating. Tiny machines, right? They're interacting with each other, tiny machines. So we're going to see internal signals and external signals that are going to influence the checkpoints. For example, that M phase checkpoint, if the kinetochores are not attached to the spindle, we're never going to see metaphase reached, and we're not going to get to move on through that. We need to make sure everybody's lined up. External signals. So introduce the idea of growth factors, right? So these could be long-distance signals or local signals from the exterior of the cell that are going to say, hey, it's time for you to grow or not to grow. And one example is the platelet-derived growth factor. Now, this also introduced another technique called cell culture. So what you can do is take tissue from an organism and chop it up into individual cells, and you can put those in a jar and grow them, grow tissue. So what you can see here is this cartoon kind of shows the example of I've chopped up tissue and put it into a cell culture tube, one without platelet-derived growth factor and one with. And only with the PDGF do the cells start to grow and proliferate. So they're looking for that external signal bound to a receptor that says, hey, the environment outside I've checked is it's acceptable. Like we're getting the signal that says we should go ahead and grow. Because you have to mimic basically the inside of a body, right? So it's looking for that signal. Now, other examples of external factors are density-dependent inhibition or anchorage-dependent. So cells, as they start to touch each other, they need to know, hey, now we're done, we should stop growing. Or, hey, I don't grow in solution, I'm supposed to be attached to something, I need to be attached to something to grow. So we see anchorage dependence. I have to let the cells settle to the bottom of the tissue culture dish, and then they would grow until they reach what's called confluence. And this is when you have a single layer of cells completely covering the bottom of the dish. And in a healthy cell culture, what's going to happen is they're going to stop growing then. They're all hanging out, being like, oh, we're all touching each other, and this is it. We don't want too many. When these things go wrong, they continue to grow. And this ultimately results in uncontrolled cell growth. This is cancer, which we'll talk about a little bit further in the future.